The overall goal of this 3D cell culture protocol is to model the glial scar event of the central nervous system using key indicator cells, microglia, astrocytes, and oligodendrocytes.
This method can help answer key questions in the neuroinflammation field, such as the potential for glial cells to enhance regenerative strategies and unravel the glial scar. The main advantage of this technique is that glial biology can be rapidly characterized in a 3D microenvironment with systematic and high-throughput manipulations.
To prepare cover slips for seeding, begin by adding 1 milliliter of 3-trimethoxysilyl-propyl methacrylate to 49 milliliters of distilled water to make a 2% solution. Then place 18 mm glass cover slips into the solution and rock for 1 hour at room temperature. After 1 hour, rinse the cover slips by serially dipping each cover slip in 3 beakers of 100 milliliters of deionized water. Then dry the cover slips in a vacuum with a desiccator and oven at 40 degrees Celsius overnight. The next day, use forceps to quickly immerse individual cover slips in 70% ethanol. Then, without drying, drop each cover slip into a well of a 12-well plate. Next, wash each well with 1 milliliter of sterile deionized water. Then add 1 milliliter of sterile polylysine to each well and incubate at room temperature for a minimum of 2 hours. Following the incubation, aspirate the PLL solution and allow the cover slips to air dry. Prepare PDMS molds by casting the premix reagents in a 10:1 ratio in a flat-bottomed polystyrene.